Welcome back friends, welcome back in another video tutorial for microbiology with Shagnik. So in this video tutorial we will going to extend our discussion on protein sequencing and assays. So from a couple of videos we are talking about protein sequencing, right? We have talked about how we can determine amino acid composition of a peptide. We have talked about how we can determine the N-terminal amino acid residue of a peptide, and we have also talked about the Edman degradation, that is to determine the amino acid sequence of a peptide. Now let me told you the process we have discussed until now, or the processes that are available in the market for protein sequencing till now, can tell us the amino acid sequence of a polypeptide with less than 50 amino acid residue. Okay, if your protein is more than 50 amino acid containing protein, then this kind of technique will not going to help you, right? And interestingly, most of the proteins are having more than 50 amino acid residue. So what will be the solution? The solution is to cleave those peptide into small peptide and then sequence them individually, right? And today we will going to learn how we can cleave them, right? Because we cannot cleave them randomly, then the sequencing will be very difficult, right? So the cleavage should be at specific position, right? So first of all we will going to use proteolytic enzyme, because we will going to hydrolyze the protein. So proteolytic enzyme, and not all type of proteolytic enzyme, but only those proteolytic enzyme that can especially cleave protein at specific location, and that is already known to us. Okay, so first of all, if we going to classify proteolytic enzyme, we will going to talk about two things. One is endopeptidase, another one is exopeptidase. Exopeptidase, as its name suggests, you will going to cut amino acids which are present at the terminus, right? And endopeptidase, as its name suggests, it will going to cut internal peptide bond. Okay, so for example of endopeptidase we can tell about trypsin, chymotrypsin, then pepsin, then elastase, and then thermolysin. And in case of exopeptidase we can talk about carboxypeptidase, aminopeptidase. Carboxypeptidase are of several types. For example, carboxypeptide A. Carboxypeptide A will going to cleave all amino acids which are present at the C terminus of a peptide except proline, lysine, and arginine. Okay, and carboxypeptide B will going to cleave arginine and lysine containing C terminus. Okay, and carboxypeptide C will going to cleave any amino acid which are present at the C terminus. Similarly, there is an important type of aminopeptidase, that is M, aminopeptidase M, which will going to cleave any N-terminal amino acid residue. Okay, now there are another type of chemicals that are also available that can break peptides. For example, cyanogen bromide. This cyanogen bromide thing is very important. We will going to talk about this cyanogen bromide in couple of topics. So this cyanogen bromide will cleave the C terminus of methionine, and hydroxylamine will going to cleave the peptide in between asparagine and lysine. Okay, so now here I have mentioned a line, that is the specificity is determined by the side chain of the amino acid on either side of the peptide bond. Why I have told you, we will going to see in this part. For example, if we going to talk about how trypsin works, or what are the location at which trypsin cleaves, we will going to say that this trypsin will going to cleave at the carboxyl side of lysine and arginine, provided that the side chain of the adjacent amino acid is not proline. Okay, so here you may find some difficulty to remember about the carboxyl side. You can just remember that trypsin will cleave after lysine and arginine. Okay, for example, let's say there is a dipeptide of lysine and glycine, so your trypsin will cleave here. Okay, similarly, let's say arginine. Similarly, let's say phenylalanine, arginine, tryptophan, then proline. So arginine here the trypsin will cleave. But if here proline is present, then it will not, right? So chymotrypsin similarly will cleave after aromatic amino acids tyrosine, phenylalanine, tryptophan. Again, provided that the next amino acid is not proline. Pepsin will going to cleave before tyrosine, phenylalanine, and tryptophan. So when we are talking about amino side, remember we are talking about that. For example, let's say this is a peptide, so here it will cleave. And again, if in place of glycine there is a proline, then pepsin will not work, right? So that is the thing. So elastase will going to cleave after alanine, glycine, serine. Again, provided the next amino acid is not proline. And thermolysin will going to cleave before any kind of hydrophobic bulky side chain containing amino acid, for example leucine, isoleucine, valine, or phenylalanine. Okay, and there is no condition here mentioned. So that is the idea, and that is why I have told you that specificity is determined by the what is present at the side chain of the amino acid on either side of the peptide bond, and we have to remember these 5 things and cyanogen bromide to solve the problem of protein sequencing that we will going to talk about in the next video. So that is the idea of specific cleavage of the polypeptide. Hope you have learned it very well, that why we are doing specific cleavage, because we the technique that is discovered until now only sequence protein with the amino acid sequence having less than 50 amino acids, right? But most of the peptides or proteins we are interested about are having amino acids more than 50, right? So what we have to done, we have to cleave it, not random cleavage but specific cleavage, right? So that we can get any idea about what type of protein or what type of amino acids present in that particular protein, right? Then we have learned that proteolytic enzyme we will going to use, that can be divided into endo and exopeptidase, carboxypeptidase, aminopeptidase. So that is the thing. And cyanogen bromide we have to remember, which will going to cleave after met hydroxylamine, trypsin, chymotrypsin, pepsin, elastase, thermolysin. So there are various type of peptides we will going to remember. So that is the idea of our specific cleavage of the peptide. So thank you for listening to this class.